suggestion what we can do, and on that suggestion he will suggest further how we can implement those works. Okay. So I'm going to present the slide here. So the initial work was in Durgapur. In Durgapur, there is a huge problem about the industrial belt wastewater treatment. Actually, though they show several kinds of regulations that they do several treatment before leaving the water in the wastewater channels, but it has been seen by several groups, not only my group, several groups have shown that who are experts in chemical science also, they have showed that these are really very much loaded with several kinds of heavy metals. Heavy metal is the problem. So here the wastewater treatment aspect is in a different way, and we are going to dealing the wastewater treatment in a different way. That is the STP based model. But before that, I am going to some of my work, what I have done. So there are several channels in Durgapur Asansol area where these type of industries, the industries are mainly steel industries, mainly metal industries. Some carbon factories are also there who uses carbon stars, those things as their product. So they leave the waste material or wastewater even without processing. Even without processing, I'm mentioning that one. In some industries, two or three big industries, they have the processing plant, but I don't know how much they are processing that element. Otherwise, they're leaving that in the channels. So with that, one of those publications, it was an area called Tamla Nala in Durgapur. It is a huge channel where total 6 or 7 industries liberate their waste material, waste water, and that Tamla Nala is going through a wide range of agricultural. Okay, in both of the sites, there are agricultural fields. So when it is going, naturally it is absorbing whatever the water have in the soil, and indirectly it is causing um, a threat to the agricultural fields around there, and we are also absorbing that one in that direction. So that was my concern. So I started work with only the lead contamination, because at that time I found a report from Bardhaman University that they showed a high lead content in Tamla Nala. I have that publication. So I started my work in that area, and I found that my idea was to find some lead bacteria, obviously, because before going to the application field, I had to check it whether any organism is degrading. Naturally, it is not degradation; it is sequestration or absorbing the chemical within the bacteria, or making it more harmless entity. Okay. So I found that there is one or two, total 3 bacteria over there who are highly lead resistant, and they are happily growing in the lead environment. So when a bacteria is happily growing in lead environment, that means they can withstand it or they can tolerate the lead. So we say it as a resistance. And then I jumped into finding the mechanism behind that how they are doing. So at that point, I found that the organism was forming a biofilm. I searched for some plasmid, whether it has some plasmid and it is making some kind of protein which is sequestering or which is absorbing lead or making trap for a lead, but I didn't find any plasmid producing such kind of enzyme. And I found they are forming biofilms, and these biofilms are very much important for in this concept because they absorb the lead and keep the lead around the bacteria or surrounding the bacteria so that the water become clean. I did some experiment and found that the lead content in the culture media where we are growing bacteria in presence and absence of lead and in presence of absence of the organism, we found that the organism is highly absorbing the lead. Even after treatment with the organism, the lead content of the solution is decreasing. Further, I tried to understand whether the organism is making some beneficial effect to the plants. So I did seed germination assay in presence and absence of the organism and obviously lead. So in the lead contaminated water, when the seeds were growing, they could not grow. Even the growth become, even some seeds were died at that concentration. But when I treated, for example, it was 100 microgram per ml or a little bit higher, up to 200, up to 200 we went. And there the organism died due to lead contamination because in absence of the lead, it grew in a very good way. And then I found when I added the organism, the lead contamination was decreased. As a result, the seed germinated in a very well manner. Okay. Then I took the organism and sequenced for that. Now, my problem was I talked in the local municipality here, municipality personnel here. They initially showed some kind of interest that yes, if you can do some kind of application over there, we can go for a pilot scale study. But the thing is that my technological barrier was there because I work in a lab. But how to apply that in the river basin or the sewage basin, that was my constraint. So at that point, the work stopped. Okay, though the proposal was preceded a little bit higher authorities. I presented there, but they were interested. But due to a technological problem, it was not fruitful in that way. So the work is there, and still now I am working a different parameter of the that portion, and I am also working in some kind of antibiotic contamination, antibiotic contaminated organism in hot spring. There is a hot spring in Bokreshwar near to Durgapur and Dubrajpuri here in Birbhum district. So that hot spring has some bacteria which is I'm currently working on. That bacteria is antibiotic resistant, and that bacteria antibiotic resistance is there, biofilm formation is there, everything is there. But the most important aspect over there is the bacteria is producing some chemical which is killing other bacteria. So whether we can use those chemical, I have studied that the chemical has. I've done mass spectroscopy too, and found that the chemical which has been secreted by the bacteria is actually protein. So there is an indication that it may be by bacteria, maybe. Still, I'm saying maybe, but it is producing that kind of thing. So we can use this bacterial supernatant or that the bacterial culture to inhibit growth of other kind of non non uh, non-healthy or pathogenic bacteria. So that's a different aspect. But today I am going to concentrate on somewhat related to my previous work. That is the Tamlanala work. And here the application is different. That is, as we know that we want to treat the water which is going through the municipality wastewater treatment plant. So in that case, I studied a little bit, and I wastewater treatment plant already use bacteria, obviously, in several of the water before entering the treatment plant and after once leaving the treatment plant is not making some difference. So that was my point of my discussion with madam that if we can add some kind of bacterial population, though it is a seed culture, but we can make some kind of repository as well suggested also in a different way. Different way means It has to be an engineering setup, obviously, where the bacteria has to be applied. The thing in our language, in our microbiological language, is called immobilization technique of the bacteria. So that is, we are making some walls or making some a place or making some packets of bacteria which will be there, I'm going to in that part also. But before that, I'd like to say a few words on the compositional principle waste as I studied. It is organic waste, 30 to 50%. So that's a huge one. Paper and cardboard, again a type of organic waste. Glasses are there, metals are there, woods are there, hazardous waste is there. So my point is that most of the part is organic waste, and that organic waste may be further. My hypothesis was that since wastewater is containing a high amount of organic waste, so after doing the physical treatment, after doing the physical filtration procedures, primary, secondary, tertiary treatment, we can further do some kind of bacterial treatment, which will either degrade the organic waste in finer particles. We can collect that and then we can utilize in other parts. For example, that may be used as some kind of nutrients for plant cultivation. If it has no toxic element, we have to taste that one. And the next part, the water part, we can use freely in the medium. So that's the thing we can do, I think. Now, the thing is that I searched for some bacterial wastewater. So, organisms here which can be applied that may be divided in aerobic, anaerobic, or facultative organisms, because once we are treating wastewater in that large amount, I think all types of organisms, facultative bacteria, anaerobic bacteria, and for the surface treatment, aerobic bacteria are also required. So some important strains I also found. Most of those are related to Pseudomonas, and Pseudomonas is very deleterious, as we know, deleterious, but it is also very much required for several types of treatment, because most of the time many bad persons have some good qualities also. So Pseudomonas, I think, in like that. Actually, Pseudomonas putida was a different story in my college. 2020, Professor Andaman Chakraborty came. I invited sir, and that was last but one seminar in his life. So sir came here, and we know all the stories about Siddhavanus Kudida and Professor Andaman Chakraborty. However, that's a different aspect. Now the facultative bacteria and anaerobic bacteria may play a good role in this type of organic degradation. Now, advantage is the efficiency of the sewage procedure. Advantage, if I say in the lab scale, after seeing the lab scale of the technology, we can apply the bacteria anywhere within the procedure, and contributes in environmental protection, minimizing the chemical use and reducing energy cost. Obviously, filtration and chemical use is which is remaining with the chemicals are remaining in the liquid or in the land over there. So that's a kind of chemical contamination towards the mother earth. So if we can reduce that, if we can use bacteria which are degrading the organic waste. So there is a treatment. This is I think all of you know better than me the procedure of this. Uh, our intention is to add bacteria very near to the just before the disinfection, maybe after clarification, before the disinfection, because after the disinfection, if we add those bacteria, that may remain as contaminant within that. So before the disinfection, we can allow the water to stay with the bacteria. That will clear the organic waste, and then we can go for disinfection. So only water will be there without the organic materials. Now, these concept is immobilized bacterial cakes actually. There the bacteria are trapped in several ways. For example, we can make some kind of meshy material, some kind of mesh we can prepare. There we can entrap the bacteria. We can use some nano particles to capture the bacteria over there, and some they form kind of interaction among them, because for bacterial activity, quorum sensing is a very important thing, as we know all. So in that case, we can adsorb the bacteria on the surface of some nanoparticles. We can self-immobilize the bacteria by interacting each with other. We can entrapment. Entrapment is exactly what I was saying, that we can create some kind of mesh where the bacteria are trapped, and we can put those type of mesh, huge type of mesh within the wastewater, where it is coming after the tertiary treatment, and even we can give some kind of nanoparticles over there. Actually, some kind of encapsulations are also used. That is encapsulated. There are sodium alginate. We do in our lab that sodium alginate beads. Those are entrapping the bacteria within them. What is the most biggest advantage of using this type of entrapment technology is that we can use those cakes of bacteria repeatedly, at least for 4 to 5 times, depending on the quality of the water, obviously, and obviously about we have to measure the efficiency of those bacteria in degrading those kind of organic waste. So we can use these cakes for several times. So that will decrease the cost. That will make the water more organic, particle free, and I don't know whether it is a very dangerous way. Even after doing those kind of organic treatment and the chlorination or something, we can use those water, if we can do in better way, for drinking water too. Okay, so if we can degrade the organic waste, actually, in my sense, the wastewater is having the organic chemical which we cannot consume. But if we can do in that efficiency, in that way, it may be converted to some kind of drinkable condition, or very close to the drinkable condition, household purposes. So it will be recycled. The water will be recycled within the society. However, advantages of immobilized bacteria, advantages wastewater treatment include increased stability, improved resistance, environmental damage, and higher removal efficiency of the pollutants. For instance, immobilized cells can enhance the removal of nutrients from sewage, significantly reduce the concentration of nitrogen, phosphorus treated water. Immobilization also mitigates the loss of active bacteria, thereby improving long-term utilization and stability of the treatment process. That I was saying, that we can use those cakes or those meshes repeatedly. So that will also decrease the cost. Role of different materials and effectiveness. It has been studied in several lab-based or semi land-based way, to whether it has been it may be applied in the field. So there are some references, and further references are also there. I have used just four over here, and this was a part of the discussion between me and Madam Dr. Donovan and Snehal Madam. So, how will we integrate this technology in the engineering perspective or currently available STPs? Okay, that's a question. That's a question. We have to think on that, and in that way, I need the suggestions from several experts in these fields. And space crunch is obviously a great problem, but I think if we think and deep that way, uh, there will have a solution. There will have a solution. Maybe some kind of engineering alterations has to be done. If the technique is established, in that case, we can apply that. USA. At that time, one of my relatives over there, he was a very aged person at that time. he told me that he designed some parts of the Manhattan water system. So at that time, he was my uncle. So he mentioned that I had to make several alterations based on the observations, as it happens in all cases that we cannot think everything sitting here and giving some lecture over there. So we have to do some kind of alterations, obviously, to introduce a new technology. So mm, uh, these was all the things, all the thoughts coming in my type of microbes like fungal or algal waste, algal compositions, only bacteria after the tertiary treatment, if it is possible.
Miss, hello. Miss, whether you have developed some consortia like that?
Yes, obviously it will be consortium, as I mentioned, that depending on the efficiency, it may be a consortium, so we can get a better result over there.
Yes. Means additional treatment is required after suppose activated sludge and all this final treatment before disinfection. This consortia is used. This is the idea, no?
Yes, sir. It will be a finer one. In my sense, it will be a finer one.
Yes, yes.
Hanumantrao ji, main purpose here is to get inputs from all of you regarding what changes can be made in the present kind of existing infrastructure facilities of sewage treatment, but the reflection is very clear from all the polluted water bodies that we have not been able to achieve even a single percent. So Kirat is he's a young dynamic person who's trying to work on this. But
Yes.
with your experiences of so many years of dealing with microbes, we just wanted some kind of an inputs from your end also, sir, that what you suggest, like in this, because see what is happening today, we are all teaching in the schools, colleges, in very much in isolation. Just we give them some sample, we isolate some bacteria, and then we make the colony, we count it, and that's all what we are teaching. But we are not doing it as an application based teaching learning. Now, with this, like yesterday, I was talking because you have done an MOU with 25 colleges.
Yes,
So from that point of view, also, that how can we come up with solution for a better kind of sewage treatment, so that results are obvious and our rivers and other water bodies can show an environmental ecological flow. So from that point of view, this discussion is. So anything additional which you can speak about what can be done with your experiences, that also would be very much welcome.
So, madam, whatever you are saying is correct. So, in the river conservation, the main problem is sewage. And if you treat the sewage in an economical way by using new technology, then it will be successful. Otherwise, it is very difficult, because the old the methods or methodology which is using, whether it is economically feasible or not. Also, efficiency, what you are telling is a very right thing, because most of this STP and all this, they are not working properly. They are not maintaining it properly, and that's why the problem is there. So the maintenance is also very important problem. And what you have suggested, this is a very important topic. Otherwise, we cannot especially the rivers and all this, we cannot purify. Also, this water is the main problem.
Sir, very basic thing which we are trying to get out of the microbiologists is that forget about ecological and efficiency and all, because we want to get a better efficacy. When we checked of the STPs, we find it is very poor in efficiency. Very poor means it's not showing any result. So that is what is the reflection what when we look at all our rivers and water bodies. So now we are trying to get from you that in so many different ways that you people have been teaching about microbiology and its applications in different ways. So in that process, you must have come across some ideas If that can be made applicable here, that we can improve the efficiency of this plants, existing plants maybe, or maybe something a very good solution which can be provided, by which we can see some change in this treatment process. Because main thing is that every day wise each one of us are producing a lot of sewage, we are contributing to it, but
Yes.
what is our contribution when it comes to giving solution for it? So from that
Yes.
point of view, I just wanted some few things which if you can add in that order.
Okay. Okay. Now, Deshmukh sir, please. AM Deshmukh sir.
I think Arvind Deshmukh ji, are you there?
Okay.
Kirat, I hope you are able to understand what I am saying and repeating.
1 minute, 1 minute.
Yes, ma'am.
Yeah,
Yes.
yeah, Deshmukh sir is here. Yes, sir.
Yes, yes. So, Kirat sir's presentation, definitely it is an addition to the older process. As Hanmantrao has told, already trickling filter is there. Many other processes are there. But in all these processes, we are dependent on a natural flora. Algae, fungi, and bacteria. Whereas Kirat sir's presentation is totally dependent on bacteria, and that too isolated and active bacteria. In short, it is an immobilization process where active bacteria are used. So definitely the process efficiency will increase. But again, I will tell you one important factor, that all these are living things. Bacteria is a living thing like you and me. So their activity depends on environmental factor. And as Hanmantrao has told, many times the processes are failure. The reason is the same. That is, either pH is not optimum, or some harmful chemicals are present in sewage, so that living bacteria get damaged. They do not work if the pH is about 10, if their pH is 4. They are active, but at adverse pH, they cannot work efficiently. And then we get a failure. So whenever Kirat sir has told, it's a very correct, but we have to look toward the environmental factor while doing the process. That is my opinion.
So now I would request Muni Rawalji to speak his mind so that we take this further.
Fit into this platform. But as a layman, I have more questions than directions to offer. And my questions are on the principles employed in the conventional treatment and whatever the research done in our lab. For example, sewage treatment, in another word, organic degradation, is the mineralization process, pulverization, not building. So the very term aerobic is out. Aerobic dependent to atmospheric oxygen is a questionable understanding. Because what if we go to organic molecules undergoing degradation 2.3 billion years ago when there was no chlorophyll and therefore no atmospheric oxygen? Or suppose if we go back to only 1 million years back, there were no industries, there were no townships, there were no sewage pipeline. Does it mean there was no organic degradation? And if it was, what it was, that remains to be understood. Dr. Kirat has done an excellent, very hard working findings. At the same time, I find certain ambiguity in that the word he has used is seed, and all immobilization people use the word seed. Now, seed comes from the available culture which can be cultivated in a lab under controlled conditions. In fact, the archaea involved in the degradation process are not cultural, with at least present day of cell and tissue culture knowledge. So the word seed is out because seed is used by activated sludge process and everybody. And still, when you say seed, you name it. You have said the word germination, you use the word germination, which is fine. But germination means you can grow under the controlled conditions. So, and anaerobic to facultative is again a misnomer, in the sense that if we provide aeration, some anaerobic forms turn into facultative. That's not right. Because most of the conventional SCPs switch on the compressor and after some time switch off. So facultative come to anaerobic. No, it's an irreversible genomic shift. Even Arvind Deshmukhji used the term algae. Algae is a higher form of cellular organization, way above archaea and bacteria. So, in spite of all the good work done world over by various members and engineers working in the sewage and waste management, now immobilization is about CN ratio 25 to 1 to 35 to 1. That is composting. Industrial waste has no organic matter, has no pathogen. I have not heard of any report where industrial waste gives rise to cholera or typhoid epidemics. No. Industrial waste can be locked down. All the industries of the world were under lockdown for 1.5 years. Sewage production, when we talk with human population, is 365 days a year, which cannot be locked down. Have we seen a single country or a town or a city observing a sewage production lockdown for 1 day? Because it's a biological need. Industry is not a biological. You can stop the compressor, you can stop the aerator. Can I stop my breathing for some time? Unless somebody is a yogi. So, if I was really looking for some process that Dr. Kirat has used and the detailing of that. Because seed increases the mass, which is a growth. And all activated sludge processes, they increase the mass of the organic matter than the incoming load. So is it a breakdown or is it addition? If you see apoptosis, that is the part that can explain organic degradation. In tertiary treatment, the nitrogen sequestration is a marriage of hundreds of microbial communities, denitrifiers, each promoting only one reaction. So NO3 to NO2 is one community, but NO2 to NO is a different community. Totally phylogenetically distant in origin. Like chemical engineers write NO3 to NO2 to NO to N2O and then to N is just arithmetic. That's not the way nature works. And all these 5 reactions which decreases the oxygen from the molecule or in the pool is a different community working under different conditions and no interchangeability. The community that can take one oxygen from NO, converting it to N2O, cannot influence nitrate to nitrite. If as a layman, if I am allowed to see my understanding, I am playing cricket, suppose, and I clean ball the batsman all the 6 times, 6 deliveries. The bowler is working hard, scientists are working hard, but not a single batsman is given out. No batsman leaves the crease. Umpire does not declare him out. Where is the problem? The problem is I step out of the bowling crease and it's a no ball. So however good length and direction and beating the batsman, if I start with a no ball, there is no gain. That's what is happening with sewage. Now we can discuss on these principles and all that, because the process will come or the experiment comes after the aim. And the principles used in the aim, we know our earlier laboratory experiments, we used to write the principle and aim and then material and method. This I'll just. And as eukaryotes, their machinery is 18S ribosome RNA. No eukaryote have any role in degradation. They eat small organisms and they grow big. But once they grow big, they increase the sludge because they will die someday. Algae die. Algae add cellulose load to the wastewater once dead. And some of the crustaceans and insects, aquatic insect species, Daphnia and all that, they increase the chitin load of the wastewater. Chitin is very difficult, very complex material to degrade. What our body throws out is not a protozoa, is not celiophora, is not algae. Algae is a very complex molecular organization with many variety of proteins and carbohydrates and lipids and vitamins and so on and so forth. Now, degradation is, can we bring down the sewage that the body rejects to a lower level of molecular organization hierarchy? So, for example, my body throws some part of partial digested carbohydrates and chiefly butyrate, which is a C4 molecule, or propionate C3, or H, which is. Now, can you bring it down to lower level of carbon hierarchy? Algae gives it to C million, C100, C1000, C100 lakhs carbon organization. That's not degradation, it's a building up process. And that term aerobic, if we see the origin of photosynthetic organisms, O2, I'll just make a statement. Our air does not have oxygen. Now let us challenge this. Dr. Kirat, this will help you perhaps.
Yes, sir.
I make a statement. Our air, atmospheric air, does not have oxygen or does not have it. Now either I am all wrong or I am right. And the problem is, what if I am right? 2 2.33 billion years ago, there was no O2 in the atmosphere. It was barely under 1%. Does it mean there were no aerobic organisms? And if there were, what were they? Or that air was different? Or that oxygen was different? So, to make the dialogue simpler, I make a statement: air does not contain oxygen. Now you can question me and I'll try to answer that. Air is O2. And O2 is a molecular, unbound molecule. Unbound oxygen. Life started with CO2. CO2 plus H2O. CO2 plus H2O, sunlight and all that. That gave us photosynthesis. Earlier photosynthesis did not go up to glucose. So it was a partial photosynthesis, which is also known as reverse Krebs cycle. So, before first atmospheric O2 appeared, CO2 is a bound oxygen molecule, bound to carbon. SO2 is bound to sulfur. FeO is a molecular oxygen bound to Fe. Nitrite, this is a bound molecular oxygen. O2 is an unbound molecular oxygen. And when you pull apart 2O from O2, it's a very high energy intensive process, which is endergonic. And degradation, organic degradation is exergonic. If you pass through a gutter and some manhole is partly open, you see hot air coming out. You see the difference in temperature. Layman. Now, if we have 6 nobles, repetitive, every principle is violated. And world over, everybody is claiming success. Bigger the city, bigger the degree of achievement. So when Dr. Snell's went to Manhattan, we had met her in some earlier sessions earlier. So I told her, kindly look into these areas. And when she questioned that, she was disappointed. Anyway, you raise more questions and then maybe my folder will open.
First of all, I would like to mention one thing to Munisa, that it was an excellent example that oxygen was there, but in bound condition. So possibilities are there. We have to leach it out. We have to take it out. Yeah.
Exactly.
Okay. So in that way, sir, what I was suggesting that we are using several kinds of algal, as Dr. Deshmukh sir also mentioned, that we are using different types of algal and bacterial, a mixture of eukaryotic and prokaryotic organisms to treat the water. So from there, I would like to extract the bacterial portion only, not only extract. Would like to focus mainly on the bacterial portion because of their faster growth rate, because of their elemental requirement, because of their diversity. That may, even I am not going in the archaea, okay, and those bacteria which are expert in degradation of these molecules. Yes, obviously that is the hexagonic process. So, which are using this bacteria, using this chemical in natural condition, we have to focus on that. That which bacteria are growing on this type of organic waste, those bacteria has to be isolated and used in the treatment process. So we have to search the nature. We have to search the nature, isn't it, sir?
Yeah, a good point. Now, what brought the lead, cadmium, mercury in the food chain? Naturally, naturally growing plant does not need cadmium or arsenic or
Okay.
mercury, right?
Yeah.
So how did it find its entry? All these heavy metals found their entry into the food chain.
Yeah.
The food heavy metals came from the industries, and industrial waste were merged with the sewage, and corporations had something to prove worthy of. So they said, okay, farmers take this. But little did they realize that with the nutrients, this sewage sludge brought the heavy metals coming from the. Now, coming to the heavy metal preference of plant matter. Some of the plants like Typha phragmites do not allow the heavy metals to enter the xylem bundle, vascular bundle of the root system. It is retained by the cortex of the root. The ectoderm of the root, if you take a cross section of the root of these plants, cortex is a mechanism to retain the lead, mercury, cadmium, strontium. It will not enter the blood vessels and go to the above portion of the vegetation, go to the seeds which we eat. But the agricultural part, that vegetation does not have that security for heavy metals. So if I eat vegetables, if I eat other food grown in that sewage, all the heavy metals will find their way from the crop to the human body. And from human bodies passes out unaffected into the waste. Some metals are picked up by the cells, and that is why we have some abnormality in our kidney and liver and all that. Microbiologists, I have a deep affinity and complaint at the same time and in same proportion. Microbiology is a tripod, and the balancing of a tripod is possible when all the three legs are in the same measure in strength. First is public health, and medical sciences and pathologists have really addressed it. Second is industrial application. Higher the growth, faster the growth, better the quality of product, better business. Nature does not care for our business. But the third and most important role of microbiology is the microbes which are linked to climate stability. All our carbon molecules we eat, carbohydrates, the lipids, they ultimately give rise to CO2 and CH4. Both are greenhouse gases. In protein containing, amino acid containing molecules, they ultimately break down and give rise to nitrous oxide, which is 300 times more potential as greenhouse gas than CO2. In the climate stability, the role of microbes, which can be the methanotrophs have an important role in some denitrifiers fungi. Unless we see the climatic stability role of microbes, I don't think microbiology can add a nanoparticle to the existing knowledge of sewage. Because sewage is a climate-linked microbiological science. Cholera and all parasites, which earlier when I was a student we were taught as protozoa, that's wrong. So, unless we have some reference and principle which can support our theory of application of principles, where this is possible. For example, Wright brothers, the first flight was barely 700 feet. But it was celebrated because they overcame gravity. ASP is an anti-entropic path, anti-gravity path in name of laws of thermodynamics. Degradation is the first law of thermodynamics. From higher potential to lower redox potential. That is degradation. A pyruvate, a 3-carbon molecule becoming acetyl coenzyme, a 23-carbon molecule to protein and to body of an insect or a crustacean or protozoa is an anti-gravity, upward flow of carbon. So is it degradation? Or is it building up a pyramid of carbon waste? Anyway, I'm just talking loudly. Dr. Snell ji came here, I was very honored. I was deeply impressed. She came with 2 of her students and stayed here with us. It is gulped when we drink water or gulp saliva or eat food, right? That O2 passes out unaltered. That O2 has no role whatsoever in digestion of the food we take, either in the alimentary canal or in the colon. So O2 is out when we talk about chemistry of or biochemistry of this. When you talk about biochemistry, of course, the enzymes and genomic constitution will be. But 2 days ago, I just sent a message to Dr. Snellji. Some officer from Bangalore, Karnataka, pollution control, and
Karyotic cellular organization.
2 prokaryotes came together, one engulfed the other, which this was a mitochondrion, a bacterium earlier. When it was engulfed by an archaeon, this gave you a eukaryotic constitution. So mitochondria and engulfing predator were both prokaryotes. Now I'm not somehow comfortably accepting that. Because if you see the membrane evolution, the phagocytic part of the membrane, no prokaryotes even today has that phagocytic membrane. All prokaryotes are stiff membrane where their shape does not change like that. So phagocytosis or amoebic kind of thing, the protozoans came in, amoeba engulfs food. So engulfing is out of question explaining the symbiosis of theory of. You see, am I putting my question?
Striking, very striking thing, yeah.
Secondly, second theory is both the prokaryotes came together and there was diffusion of DNA material. And therefore nucleus and all that kind of thing. But then I have a problem there. The ribosome complex with prokaryote is 16S constitution. If 2 prokaryotes come together, it should be 32S or 2 into 16S. But how it gave rise to 18S, which is a prokaryotic ribosome complex? That 16 to 18 needs to be understood or first explained by something. Otherwise, the entire symbiosis theory goes for a toss. And when Dr. Snellji took me to microbiological session some 3-4 months ago, a very learned scientist, she had worked on this 16S ribosome structure, and it's a wonderful lecture. But when I posed this question, she was shell shocked. I said, माफ करो भाई, मेरी कोई गलती हो गई, कुछ irrelevant topic पूछा. She says, doing this for last 20 years and why it did not strike me that prokaryotes are not amoeboid, not phagocytic in their membrane culture. So मैं तो पढ़ता रहता हूँ और सवाल ही उठते हैं, मेरे पास कोई जवाब नहीं है. जवाब एक ही है कि bring your sewage treatment plant close to the colon, because the sewage has its origin from the colon. Forget about industries, you can lock down the industries. 300 years ago, there were no industries. There are only humans here. And now, before humans came, there is the vegetation, foliage. Animals die in the river and sea. So their dead cell contribute to the sewage production. Why are we linking sewage to that something that emerges out of a pipe? Even a leaf falls, or cow dung falls on the ground, then it mixes with the soil, some atmospheric moisture, this, that, air. And if aerobic can solve the problem, why are the hill mounds world over are catching fire? All wastewater hills piling up, they catch fire in monsoon. And they are exposed to the air ever since their birth. So I'm only, as a layman, unless I see the principle. 18S category is the feature of the endoplasmic membrane after the compartmentalization. So if we understand membrane well in its evolution, I think we have the key, because all 18S creatures, they later on engage with O2 and etc. etc. So, and fine, thank you. You can, if you come across any reference, I'll be happy.
Different thought procedure. Now I have changed.